This lecture is on the saccule and utricle. So the saccule and utricle then are suspended in perilymph. You remember that fluid. So here we have perilymph again. And they are connected to each other by a small duct. And the saccule also connects with the cochlea and the utricle also connects with the semicircular canals. So there's a lot to understand here. Again, this is just going to be strictly a diagram, but we have the utricle here. And here I'm going to attempt to draw the semicircular canals. It's not that pointy. So here then we have the semicircular canals. And here we have the utricle. And then here, connected right up to it, then here, then we have the saccule. And then coming off of that is this great snail-shaped tube, which I am not going to try to draw as tightly wound as it really is, but the point is we have this great spiraling cochlea deep inside us, which is ordinarily all the way tightly wound like this. That would be hard for me to draw as a tube, but then this then is your cochlea, and more on these other things later. So what they do, now speaking of the saccule and the utricle, then these house equilibrium receptors that are called maculae. That's a C there. Called maculae. So, as it sounds, these detect equilibrium. And this means that they detect the position of the head with respect to gravity. What did I just say? What I am basically telling you is that these receptors here, these maculae, they tell you where down is. Specifically, they tell you where down is with relation to your head. So, as I said, they detect the position of the head with respect to gravity, that is to say, where down is. And related to this, then, is that in addition, they respond to what are called straight line changes in speed and direction. But not rotational movements. Okay, what did I just say? It goes like this. If I'm standing here and I'm now going to make a change in my speed, I just did that. That would have been picked up by my maculae. I was walking in a straight line and my speed suddenly changed. I stopped, I started, I stopped again. Another thing that it could do is it could detect slight changes in direction, or actually major changes in direction as well. So the point is, if you are walking forward and you suddenly change your direction, the maculae will pick that up as well. So they will pick up straight line changes, stopping, starting, slowing down, speeding up, and they will also detect if you turn. But not rotational movement, which is to say this, or this. Is this making sense? So what I'm going to do now is draw you a couple of pictures, and then I will take quite a bit of time explaining what those pictures are showing you. So bear with me for a moment here. I'm going to draw you a macula. So I'm going to start then with some little black rocks. Here they are. When I say rocks, I'm not joking. These are what are called, I think I'm going to give myself a little room over here maybe. So these then are what are called otoliths. Now when you see lith, you know you're dealing with a rock, okay? These rocks sit on a membrane. Here's the membrane, and it has kind of an interesting shape to it. So this now, and I'm just going to make it a little bit textured so you can kind of see it here. This then is the otolithic membrane. And let's use some other colors so you can see things a little bit better. So then we have a bunch of little hairs, like so. I think I'm going to shorten those up. So here we have all these little hairs. Which are grabbed onto by this structure here. Go. Here we have these structures. And these now, I'm just going to indicate two of them, but these then are your hair cells. That's what they're actually called. And then surrounding the hair cells are a bunch of support cells, which I'm just going to indicate here with some red, so you kind of know where they are. Here then we have our support cells. Like so. So these now are support cells. And what happens then is that these hair cells are connected to nerve fibers. So here then we have the nerve fibers all heading off like so. So here you have nerve fibers. And what you are looking at here then is a picture of a macula when the head is upright. So when your head is upright, you have these little rocks there that are sitting on this membrane, nicely balanced, nicely supported. So that then is your head upright macula. So now, when you have your head sideways, let's do this. Here we have our otoliths again. Remember, when you see the word lith, it has to do with rocks. And here they are, sitting on their membrane. Here's the membrane again. But things have gotten a little bit different here. This is where things get a little bit tricky to draw, so bear with me here. So here, again, we have the otolithic membrane. But what's different now, let's take a look at those purple hair cells. Is that here, these poor hair cells here, this has moved down. So these now are stretched way up. You can't even see that. There we go. And then here, so then here we have the hair cells. Here we have the nerve fibers. And then, of course, around all of this, we still have the support cells. So the support cells are still going to be in here, roughly speaking. So what I'm getting at here is that this person's head is sideways. And so all of these otoliths then are going down towards gravity. When your head is sideways like this, all of your little otoliths are then heading down towards gravity. Here they were nicely supported. Here they're not. They're falling down. That stretches these hair fibers then. And hang on, I need to label that, don't I? These, as before, then are your hair cells. There we go. But the point is that as you have your head sideways and the otoliths are going down towards gravity, then that is going to pull then this down and pull the hair cells down with them. These, meanwhile, are embedded in the support cell. So as a result, you get some hair stretching and some hair bending here. And that is the movement, this bending of these hairs then, that tells you that your head is sideways. It tells you that your head has changed its orientation with relation to gravity. It has changed its orientation with relation to down. So now let's talk about how this works. We have these two nice pictures here. There we go. So here's how it works. The hair cells are sensory receptors. And when the hairs move, this creates a nerve message. And the hair cells, as you can see, are scattered among support cells. And the hairs on the hair cells project outward. And the hairs are embedded then in the otolithic membrane, as I have drawn here and as I have drawn here. And the otolithic membrane then is studded with tiny stones, actual rocks here, that are called otoliths. Lith means rock, oto means having to do with the ear. So these are literally ear rocks or ear stones. And the otoliths are heavier. That is to say, they are more dense than other parts of this arrangement. So as you can see now, you're beginning to see how this all works. So this means that the otoliths go down, that is to say, towards gravity, when possible. Is it possible here? No, they're just sitting there when your head is upright. But when it is possible, like when your head is sideways, this bends the hairs. Which creates a message which is sent to the brain. And as I have indicated, this tells us the position of our head. In relation to gravity. In other words, this tells us where down is. Because remember, my head could be upright, it could be fully on its side, in which case gravity is there. My head could be only slightly tilted. Gravity is still here. No matter how my head is tilted, those otoliths are going to be going down. Sorry, it's hard to do this on camera. The otoliths are always just going to be going like this, wherever that is. So as I said, when I'm standing straight up, the otoliths are supported, head on side, otoliths are going down sideways, but no matter how my head is tilted, those otoliths are going to be going down, and that will be bending the hairs accordingly. So I also told you about starting and stopping and directional changes. So the otoliths then also bend the hairs when we, let's put it over here, when we start moving, stop moving, or change speed. That is to say, slow down or speed up. And getting back to the overall structure here, the utricle and saccule, remember they are suspended in fluid, but they are also filled with fluid. And the utricle and saccule then are both filled with endolymph, which is a fluid. And just to remember one from the other, remember endo means inside, whereas peri means around the outside, like the periphery. So that's how you can remember that. And that is it for the utricle and the saccule.